Okay, well, I guess I'll make a start. So, if anybody had any doubts, I'll be giving this lecture in English. I put the slides up onto the UN cloud this morning so you can get the PDF of the slide set. And generally speaking, every time I have a reference, that should be a clickable link if you ever want to check out the papers and find them. Okay, so the teacher kindly asked me to give this lecture and said, can you do something about persistent viral infections? And of course I said, oh yeah, I can do that, easy, yeah, I'd love to. But then I realized this was actually going to be a lot of work because that means if you take out HIV, that means what are persistent viral infections? You're talking about chronic hepatitis viruses, human papilloma viruses, and all the herpes virus infections. And I decided to leave out the polyoma viruses because in terms of overall health impact, they're a little bit less important. So really that's what is going to be covered in this lecture. And I'll start with a kind of reminder about persistent viral infections. So, when you get infected by some kind of pathogen, your immune system will mount a response. And there are going to be three possible outcomes. Either the immune system manages to eradicate the pathogen, in which case that's it, you win. Or your immune response is not able to control the acute infection, then you'll get more and more severe symptoms, and then you end up perhaps, dying. That's a win for the pathogen. And the third case is this one in the middle here, where the immune system can't eradicate the pathogen, but it does control it to some extent. So you get a persistent infection. And that's kind of a draw, right? Okay, and that's what we're talking about. So in persistent infections, there are really two types. There are latent infections and chronic infections. So in a latent infection, after a symptomatic primary infection, then there's a phase of where it seems like the immune system has cleared the pathogen, so there's no active viral replication that can be detected. But in fact, the virus is still there in a kind of refuge site, latently infecting some cells someplace in the body. And then years or months or decades later, you can have periodic episodes of reactivation, which are accompanied by symptoms. Now, contrary to that, you have chronic infections. And in a chronic infection, you always have active virus replication all throughout the duration of the infection. And that means that, the virus is never cleared from the blood. And you've got chronic, continuous replication, symptoms that are continuously present, tending to get worse and worse. And then as time goes on, now this can lead to pathology and death. So, the big chronic infections are chronic hepatitis B, hepatitis C. The latent infections are really the masters of this are the herpes viruses, herpes simplex virus 1 and 2, varicella zoster virus, cytomegalovirus, and Epstein-Barr virus. And somewhere in between, you've got HIV and the papilloma viruses. So in HIV infection, you have latently infected cells. This is the reservoir. But of course, in an untreated infection, then you've got continuous virus replication. In papillomavirus infections, I kind of put it halfway between latent and chronic, as we'll see why. Now, in order for all of these viruses to persist and continue, infecting us for years or decades, they've all got to dodge the immune response. So they've all got to have some mechanism to escape from the innate response, from the cell-mediated response. I'll mostly be talking about T cells. I don't know why. There's just something about NK cells that I don't like, okay? So that's a bit of a blind spot for me. And they've all got to avoid the immune response, got to avoid neutralizing antibodies. Now, why should we care about this? Why is this important? Well, one of the main reasons is several of these viruses cause, they're quite important causes of cancer. So hepatitis B, hepatitis C, they're related to hepatocellular carcinoma. Epstein-Barr virus causes a number of B cell and other malignancies, and papillomaviruses, or some papillomavirus genotypes, cause cervical cancer and oropharyngeal cancer. So these are important pathogens with quite a high disease burden associated with them. And their ability to maintain a persistent infection is really behind their kind of carcinogenic potential. Okay, so what I'm going to do is, for all of these viruses, go through, how the virus can, what are the particular immune mechanisms that are engaged and how these viruses can dodge them. Okay, so starting off with the hepatitis viruses. So if you kind of look at the overall incidence of chronic hepatitis B and C in the world, this slide is a couple of years old now, but it was estimated, maybe this is about like 5 to 10 years old, that there might be about 325 million with a chronic hepatitis virus infection. And the main regions of the world that are affected, East Asia, Sub-Saharan Africa. And this pretty much can be superimposed on the incidence of hepatocellular carcinoma. So these two viral infections are, the main drivers of this cancer worldwide. So if we look at hepatitis C, more recent data, the estimate is that there are maybe 50 million people who are chronically infected. It's about 0.6% of the world population. In France, a little bit less, maybe 130,000 people with chronic hepatitis C infection. And worldwide, that would be maybe 71 million people, with the Middle East and North Africa having the highest prevalence, then maybe East Asia. There are 6 genotypes of the virus, and with kind of slightly different geographical distribution. The genotypes were described and numbered in the States. So of course, genotype 1, that's the most prevalent virus in North America. And then it goes like this, 1 to 6. But what kind of a virus is hepatitis C? It's a member of the Flaviviridae family. So these are single-stranded, positive strand RNA viruses with a genome of about 10 kilobases, with an envelope particle with an icosahedral capsid. Transmission is through blood. That's really the major mode of transmission. Exposure to blood products, or things like tattoos, dental treatment, medical treatment where the instruments aren't really correctly sterilized from one person to another. Mother to child transmission does occur, but it's relatively minor mode of transmission. Sexual transmission does occur, but only in the case where you've got some kind of exposure to blood. And this virus will infect hepatocytes, and there's some evidence that it can infect B lymphocytes. So once you get infected by hepatitis C virus as an adult, then maybe 15 to 20% of people will be able to eliminate the infection. You'll get spontaneous resolution, but maybe 80, 85% of people who are infected go on to a chronic infection. And if you have a chronic infection, when it's untreated, then there'll be a progression of chronic infection, which will lead to hepatic fibrosis, cirrhosis, and the end stage is hepatocellular carcinoma. Now, as I said, untreated infection, the median time from infection, although we don't always know the date of infection, to cirrhosis is about 30 years. And overall, someone who has chronic hepatitis C have about a 50% risk of dying from their hepatitis C infection because it takes so long. The other 50% will be, heart attack, car accident, this kind of thing. The rate of progression of disease isn't constant. So the older you are, the faster you'll have a disease progression. Male sex, alcohol consumption, HIV co-infection are correlated with faster disease progression. So fortunately, in France, there's a curative therapy. So there are direct acting antivirals. And the current recommendations in France, I believe, are either an association of glicabovir and pibrentasvir, 8 weeks course, or sofosbuvir and velpatasvir, 12 weeks. And both of those treatments will give like a 90 to 95% cure rate, independent of the genotype. So cure rate in hepatitis C, what does that mean? That means sustained virological response. So after the course of therapy, people finish their therapy, there'll be no more virus detectable in the blood, and then 1 month, 2 months, 3 months, 6 months afterwards, there's still nothing. So the virus has really been eradicated. Okay, so imagine we go back in time 20 years ago, or go to a place in the world where you don't have access to this kind of therapy. How is the virus maintaining a chronic infection? Okay, first thing is got to dodge the innate immune response. And there are 2 viral proteins that are involved in this. So here's the genome of hepatitis C virus. So it codes for 1 single polyprotein, which is going to be cleaved into the individual functional proteins by the viral NS3 protease. Now the NS3 protease can also cleave MAVs and TRIF, which are essential signaling intermediates in RIG-I, MDA5 signaling, and toll-like receptor 3 signaling. So, innate immune receptors for detecting RNA, which makes sense. It's an RNA virus, right? So inactivating the host detection of unusual pathogenic RNAs makes sense for this virus. Now, the other major mechanism by which hepatitis C escapes from the innate response is NS5A, this protein which blocks JAK-STAT signaling and inhibits the RNA-dependent protein kinase. So both of these are important elements in interferon signaling, type 1 and type 3 interferons mainly. Now, the importance of NS5A is really demonstrated by the effect of NS5A inhibitors. So all of the Tasvir drug family that we saw before, this pibrentasvir and velpatasvir, they're in NS5A inhibitors. So what happens if you just inhibit this one protein? So we have to go back in the literature to look at the phase 1 data when these molecules were tested as monotherapies. So here's data from 2011, phase 1 trial of the first NS5A inhibitor. You're just giving this drug twice a day, and for a genotype 1b infection, you get like 4 log reduction in viral load over 1 week, 2 weeks. And that's just by inhibiting this one virus protein. So if you stop the virus from dodging the interferon response, that's really having a huge effect. Okay, so this is the major mechanism by which hepatitis C is actually persisting in the body by this, making the virus resistant to the effects of interferon. How about the CTL response? Well, there's good data that the CTL response is important for viral clearance because there are strong associations between certain HLA class 1 alleles and spontaneous clearance of the virus. 20% of people who get infected will be able to eradicate the virus, and this is dependent on a good CTL response. So how does the virus avoid this? Well, it's an RNA virus with a high viral load, so it's continuously producing escape mutants. And the selection of CTL escape mutants has been documented in primary infection. So this is an important mechanism for the establishment of a chronic infection, hepatitis C, just this kind of CTL escape by producing mutants that are either, affecting CTL epitope processing and presentation, or binding to HLA class 1 molecules, or just the interaction with the TCR. How about antibodies? Because if you develop neutralizing antibodies, maybe you could clear out all the circulating viruses. Well, once again, you've got a high mutation rate in this RNA virus, so you're always inducing new mutations in the viral envelope glycoproteins. Another mechanism that's important is this thing called a glycan shield, similar to what you see in HIV. These, or hepatitis C envelope glycoproteins, are highly glycosylated. So neutralizing epitopes can be kind of, covered and shielded by these N-linked glycans. So it's very difficult, or rather more difficult, for for antibodies to get access to these sites on the protein that are important for binding and infecting hepatocytes. Now, another aspect to the glycan shield is that these N-linked glycans are added onto asparagine residues. And so if these asparagine residues get mutated, then it will change the glycosylation sites. So, if the immune system manages to develop an antibody that can recognize perhaps this epitope with a part of this glycan here, the virus changes its glycosylation sites, and that will change the epitopic landscape of the envelope glycoprotein. So that's an important mechanism of escape from the humoral response. But perhaps even more importantly is the ability of hepatitis C to have cell-to-cell transmission among hepatocytes, at the tight junctions. So, the virus doesn't have to be released into the blood and then reinfect hepatocytes in the liver sinusoids. It can just go from one hepatocyte to the other, and this is much less susceptible to antibody neutralization. And this was shown in experiments, from more than almost 15 years ago now. You can test monoclonal neutralizing antibodies either for cell-free infectivity, so neutralizing virus pseudotypes. So you can see for this one, 10 microgram per ml, the black bars here, almost all the infection is inhibited, same for this monoclonal. But at that same dose, if you've got co-culture transmission, it's not blocking all the infection. So this is an important mechanism of dodging the immune response. Okay, so how about hepatitis B? So once again, an important cause of liver cancer, and more people infected worldwide with hepatitis B than with hepatitis C. So once again, the figure is a little bit out of date, but the data from this year, the estimation is about 254 million people with a chronic hepatitis B infection. And that's, it's not negligible. It's about 3% of the world population. And in terms of prevalence in France, 10 times less. Now, there are different genotypes of hepatitis B virus. with different distributions in the world. So here we've got A2, D2, D3, Sub-Saharan Africa, A1, D3, D6. And the places where you have the most seroprevalence are Sub-Saharan Africa, Eastern Asia again. Not the same as for hepatitis C, okay? Hepatitis C, it was the Middle East, okay? Here we have two other regions in the world where the hepatitis B prevalence is having a bigger effect on public health. So what is this virus like? It's, okay, part of the Hepadnaviridae family, which are reverse transcribing viruses. Okay, but unlike retroviruses, in the virus particle, you find DNA, you don't find RNA. And the genome is very small, 3 kilobases of partially double-stranded circular DNA. The infectious particles look like this. So you've got an icosahedral capsid and an envelope. Transmission, once again, blood, but this time hepatitis B much more easily transmissible than hepatitis C. Sexual transmission is possible, and the main mechanism of transmission is mother to child. Worldwide, this is the most important thing. Infects hepatocytes, and once again, once you get a chronic infection, then you have this disease progression from chronic infection over several decades, cirrhosis, and then carcinoma. Now, whether you get a chronic infection or whether you eliminate the virus depends a lot on the age at infection. So in adults, I don't know, maybe 80–85% of people will eliminate the virus, and only a minority of people will develop a chronic infection. But at perinatal infection, then this ratio is inverse. So almost all perinatal infections will go on to become chronic. And the mechanism is perinatal, it's not in utero, it's during childbirth. The baby gets in contact with infected blood from the mother. So once again, same cofactors for disease progression as hepatitis C: age, male sex, alcohol consumption, HIV co-infection. And there are antiviral drugs available for therapy. So because these are reverse transcribing viruses, then reverse transcriptase inhibitors can be used. And these are used as monotherapy, either entecavir or tenofovir. No real clinical or virological benefit in associating more than one antiviral. Okay, we'll have a look at why this can work in a minute. Now, one thing to remember here is that in contrast to hepatitis C, hepatitis B antiviral therapy is not curative. So the aim is to suppress the viral load to slow down disease progression. But if you stop therapy, there's a viral rebound, just like in HIV infection. So this is one of the aims in hepatitis B therapy now, is to go work towards curative therapies. Okay, so the innate response. How does hepatitis B avoid the innate response? Now, what we know about hepatitis B infection is that it seems to go undetected during primary infection, and this we know this because of experimental infections in chimpanzees. Now, I must admit, I kind of hesitated before putting this data up here because personally, if I find this experiment ethical, it's about the same thing as, you know, experimenting on a prisoner that you've got in a cage somewhere. They can't give you consent, you just do something to them. But nevertheless, scientifically, it gave us some important information, so I'm going to present it anyway. So here we've got experimental infection of chimpanzees with hepatitis B. So what you have on the graph, so this is weeks after infection. This curve here is the viral load. The bars are the capsid antigen in peripheral blood, and the white well, round white circles here, this is serum alanine transferase. So this is liver damage. So what you can see here is that, the peak of virus replication isn't really what's causing the liver damage. Okay. But what's happening here is you've got CTL infiltration, and then this is what's bringing the viral load down, but it's causing the hepatitis, the acute hepatitis. So this animal clears infection, but it's got acute hepatitis, which is caused by the immune response. So if you look at, and you can see that this is T cell infiltration, because in these liver biopsies, this is where you've got CD3 on a northern blot. Okay, so this liver damage here coincides with interferon gamma and a T cell infiltrate. One interferon sensitive gene, one ISG, is oligoadenylate synthase. You can see you get a bit of an innate immune response here, 1 month after infection, but mostly, the interferon response is getting activated when you have the T cells infiltrating into the liver and secreting interferon gamma. Now, this kind of finding has been recapitulated in chronic infection in people, and I thought this was really clever experimental setup here. So they've got liver biopsies from patients with chronic hepatitis B or some other liver pathology, HBV negative, and they get the biopsy in a kind of like, a carrot from the needle aspirate, and they cut it up into little bits. And they put each of them into a tube with cell culture medium within about 1 hour after the biopsy. And then they culture these liver, these small liver biopsies over 3 hours and 6 hours in the presence or in the absence of poly-IC to stimulate an interferon response. So without any stimulus, okay, there's no interferon beta released in these cultures. So you've got a chronic hepatitis B infection, there's no interferon beta being released at all by these infected hepatocytes. Now, the question here was whether the virus is actually preventing interferon beta from being released. So can these infected hepatocytes actually produce an interferon response? So that's what they saw when they added the poly-IC. And here you've got pretty good interferon beta response in the chronic hepatitis B infected biopsies, just the same as in the control biopsies. So in fact, there's a normal interferon response in chronic hepatitis B infection. The virus isn't really, doing anything to inhibit the interferon response. So the interpretation here is that, that hepatitis B is a kind of stealth virus. It goes undetected in infected hepatocytes. And the reason that we think this can happen is because the viral life cycle avoids producing kind of unusual DNA or RNA structures in the cytosol. So after infection, you get the capsids transported to the nucleus, which then release the DNA into the nucleus. The partially circular DNA will be repaired by cellular enzymes, and then you get expression of viral RNAs, which, biochemically, they're identical to cellular mRNAs. So they don't activate any innate pattern recognition receptors. Now, in order to reproduce an infectious particle, these messenger RNAs have got to be reverse transcribed into DNA. And that would activate some kind of innate immune receptors. However, the reverse transcription occurs within the capsid, okay? So it's not accessible to any kind of host cell pattern recognition receptor. So this is really pretty clever, and that's why people think, hepatitis B doesn't really activate any immune response. Okay, how about the humoral response? Now, what everybody should know about hepatitis B infection is that there is a relationship between seroconversion and chronic hepatitis B. So if you get infected and resolve the infection, that is, eliminate the virus, then you will develop antibodies to the HBs antigen. So this is the envelope glycoproteins, the HBc antigen, the capsid antigen, and the HBe antigen, which is a short peptide at the N terminus of the capsid. So you get full seroconversion to these virus structural proteins, and you don't find any circulating glycoprotein, HBs antigen, or hepatite or HBe antigen. Now, if you get partial seroconversion, you seroconvert to HBc and HBe, then you'll have persistent HBV antigenemia. So you'll have the presence of HBs antigen, but no HBe antigen. So this is a chronic infection, but it has low risk for a progression to hepatocellular carcinoma. Now, the worst thing is if you've got seroconversion only to the HBc antigen, in which case you'll have no antibodies against HBs or HBe, so both of these antigens are found in the circulation. And this is what's associated to the higher risk of disease progression. So this failure to seroconvert to the surface glycoproteins really is what correlates with chronic infection. And well, one of the things that and the basic idea is that, you know, with people with a chronic infection, if we could induce the seroconversion, then maybe they'll be able to clear the virus and they'll be cured. However, you know, if you vaccinate people with a chronic hepatitis B infection, they won't seroconvert. So they have a specific immune tolerance to the HBs antigen. So if you know the hepatitis B vaccine, it's a recombinant protein vaccine of the HBs antigen that forms spontaneously virus-like particles. So the immunological efficacy is very, very high. Everybody is vaccinated will get a very good titer of anti-HBs antibodies, but someone with chronic hepatitis B will not. So what is happening here? Another thing that's really, I think, related to this is that in the blood of someone who's got chronic hepatitis B, you don't just find infectious particles. So you also find a lot of empty particles which don't have capsid antigen. So what you can see here, I don't know where this is actually from, it's maybe purified from somebody's blood. So these round spherical type particles here, these are the infectious particles, okay? They've got capsid antigen, they've got the viral DNA inside them. But all of these kind of tubular pleiotropic particles here, these are just formed with the HBs antigen, the virus glycoprotein, membrane or an envelope formed from the cellular plasma membrane, and there's no DNA included. And there's a lot in the blood. So I think what we have here is we've got a phenomenon of high dose tolerance. And if you think, put yourself in the place of an antigen presenting cell, you're going to be taking up a lot of these empty HBs particles. There's no DNA, there's no RNA in them. So they're not associated with any PAMPs, okay? And that's related to this specific humoral tolerance to HBs antigen. Now, this kind of mechanism used by hepatitis B to escape from the humoral response is really pretty specific. And one of the reasons is because there's not very much genetic variability in the envelope glycoproteins of hepatitis B. Now you might think that's kind of strange because it's a reverse transcribing virus. So that means that the reverse transcriptase will generate errors, an error-prone transcriptase, so you would expect a high mutation rate. Now and it's true, there is that high mutation rate, but what is limiting the incorporation of escape mutations into the HBS antigen is the fact that the virus has got a very, very small genome. It's only 3.2 kilobases long, and it's got to code for a reverse transcriptase, capsid antigen, and the envelope glycoproteins, these S antigens. And the only way it can code all of this in such a small molecule is to use the same DNA sequence to code for the reverse transcriptase, so that's P, the polymerase, and the surface glycoprotein S. So the same stretch of DNA has got to code for two very different proteins. And that means that you can't really support a lot of mutation in the HBS antigen. So that's why there's really no escape from neutralizing antibodies. Okay, it's not like hepatitis C, it's not like HIV. And there's not very much variability between genotypes. That's why the vaccine can be used all over the world. Okay, so this is some aspect of fundamental virology that has an important consequence for the antiviral immunity. And this is also the reason, why antiviral monotherapy can be used. Okay, so entecavir or tenofovir can be used as monotherapy, and that's because the reverse transcriptase is under the same kind of functional constraint. So if the reverse transcriptase integrates a mutation that will confer resistance to one of these antivirals, it's also going to have an impact on the surface glycoprotein and have an impact on the fitness of the virus. So it means it's got a big genetic barrier to all these escape mutations because they have an impact on fitness. So that's why monotherapy can work for hepatitis B, but you can't use it for hepatitis C or HIV. Okay, the virus is kind of not activating the innate immune response. It's got this mechanism to prevent induction of antibodies. How about T cells? So this is just results from one paper a few years ago now, where they looked at the number and the phenotype of HBV-specific CTL using tetramers, HLA-A2 tetramers loaded with epitopes from different HBV proteins. Okay, the capsid antigen, the envelope HBS antigen, and the polymerase. And they're comparing patients with acute hepatitis B and chronic hepatitis B. And of course, most adults with acute hepatitis B will eliminate the virus. So this is what they found, okay? So if you look at someone with an acute hepatitis B infection, you'll get really quite a reasonable amount of CTL, 0.6% of the CD8 cells, 0.2, 0.1. If you add these up, you've got maybe minimum 2, 3 or 4% of all the CD8 T cells antiviral CTL during the acute infection. During the chronic infection, these are much lower, okay? And they couldn't find any HBS antigen-specific CTL. So this kind of is consistent with the idea that you have some specific immune tolerance against HBS antigen, but there's also a lot fewer CTL against the other viral antigens. So what is happening here? Well, the idea is that this is probably related to the kind of tolerizing effect of the liver environment. So as you may or may not know, you can transplant the liver without any HLA and it will not be rejected. So that's an indication that the liver can tolerate the T cell response, or at least won't be eliciting a strong T cell effector response. And the reason this happens is because of the particular relationship between hepatocytes and the liver sinusoids. So the liver sinusoidal epithelial cells, or endothelial cells, excuse me, are fenestrated. So hepatocytes poke up some kind of pseudopodia up into the liver thyroid. And so T cells that are passing through here, they can interact with epitopes presented by HLA molecules, by hepatocytes, without having to pass through the endothelial membrane. That would normally be dependent on T cell activation, right? But here, you've just got non-activated naive T cells. They're going to be interacting with antigens presented on the hepatocyte membrane in a non-inflammatory context. And so this is probably going to result in, T cell dysfunction tolerance. Now, the authors of this very excellent review from a couple of years ago, they said that, you might be able to get priming of the T cell response by Kupffer cells. Not really clear to me why they think that the priming is happening in the liver or not in the secondary lymphoid organs as a result of, the high viremia. But in any case, this direct interaction of T cells with antigens presented on hepatocytes in the absence of inflammation seems to be an important mechanism in having a non-optimal T cell response against the virus, and specifically, particularly in children. Okay, so if you don't have any humoral response to HBs antigen, you have a chronic hepatitis B. So the idea is that, if you can induce HBs seroconversion, then people might be able to eliminate the virus and be cured of their chronic hepatitis B. So there are, a lot of teams working on this. And I think there are basically three types of approaches of immunotherapy to try and stimulate the immune response against HBs. So one of them, try TLR agonists to try and, break the immune, silence of the infection here. So there's one clinical trial, TLR8 agonists plus nucleotide antivirals. So it's adjuvant therapy together with antivirals. Run for 6 months, 1 injection per week, 48 patients, 39 in the treatment arm, 9 in the placebo arm. And what are the results? So they got HBs antigen cleared in 2 out of 39 patients in the treatment arm, 0 out of 9 in the placebo arm. Another possible therapy is checkpoint inhibitors. You're thinking, if you've got some kind of immune tolerance, maybe if we add a checkpoint inhibitor, that will allow the immune system to, or virus-specific T cells to, not be, blocked in their development. So there are several trials on checkpoint inhibitors. Here's one published this year. Anti-PD-L1, once again with antivirals, 6 months therapy. Dose response, dose escalation kind of therapy, 60 with the 1 milligram dose, 59 with the 2.5 milligram dose, 30 with placebo. 30 with placebo. This 9 has come over from here, okay. And then they got clearance, only 3 out of 10 patients with the 1 milligram dose, and only those with the lowest baseline viral load. And if they just looked at everybody, the 2 treatment arms and the placebo, this is the change in viral load. Lower, reduction by about 0.3 logs, and no dose effect with the drug. So if you just compare this with the hepatitis C and the daclitazvir NS5A inhibitor, we had like a 4 log reduction in 2 weeks. So that's really good. This is a 0.3 log reduction in 6 months. It's not so great, okay? It's not a great response, virological response. And the last kind of therapeutic approach is therapeutic vaccination. So once again, recent study from this year, and they're trying different types of viral vectored vaccines. So modified Vaccinia ankara, so Vaccinia virus, recombinant Vaccinia virus twice, or recombinant adenovirus plus a Vaccinia, adenovirus plus vaccinia with nivolumab. So this is, what is that, anti-PD-1 or anti-PD-L1? Everybody? Okay. Who says nivolumab is an anti-PD-1? Who says nivolumab is an anti-PD-L1? It's an anti-PD-1, blocking anti-PD-1. Okay. So they're trying these different associations of therapeutic vaccination with plus or minus checkpoint inhibitor. So this is the best result they got here, this one. Okay, some reduction in viral load in treatment arm 3. Okay, but once again, only a few people, 2 out of 55 patients, actually cleared the virus. So, you know, we're at the beginning of this type of therapy. You get some sustained virological responses, but not very many. Okay, so this is, I think, a big active area of research for chronic hepatitis B infection, working towards a curative therapy with, these immunotherapies. Okay, so we've got about 40 minutes to look at our other 2 types of viruses. So first, we'll be looking at human papillomaviruses. Okay, so once again, human papillomaviruses are the causal agents of cervical cancer and some oropharyngeal cancers. Worldwide, 4th leading cause of cancer in women, and maybe about 660,000 new cases, 350,000 deaths per year worldwide. And cervical cancer is exclusively caused by high-risk HPVs. So it's a sexually transmitted disease that gives you cancer, okay? And the most affected countries, sub-Saharan Africa, maybe Australasia, South America, not so much Europe, not so much the States. Now, HPV, human papillomaviruses, you always have to remember this isn't one thing, okay? There are about 200 distinct species of human papillomavirus that are divided into 3 genera. So there are alpha, beta, and gamma papillomaviruses. And the ones that are associated with cancer are the alpha HPVs. So HPV 16, 18, 45, 33, these are high-risk alpha HPVs. Now, not all alpha HPVs are associated with cancer, but all cancer-associated HPVs are alpha. Okay, if everyone understands what that means. Okay, so what is happening? So what are papillomaviruses? So these are small double-stranded DNA viruses with about 8 kilobase circular DNA genome. The viral particles are icosahedral non-enveloped, looks like this. Actually, these are VLPs that we made in the lab. And transmission of the high-risk alpha HPVs is sexual. Okay, so you get transmission by unprotected sex, and they have tropism for the basal epithelial cells in the mucosa. Now, what happens once you've been infected is you'll have a progression of the infection from, just a few cells infected at the mucosa. Then you have low-grade intraepithelial neoplasia, higher grade dysplasia, and then maybe it will progress to invasive cervical cancer. Now, at each stage of this progression, you can develop an immune response and eliminate the virus and the infected cells. So these infections or the cancer, progression to cancer is low penetrance, okay? So if you go for a checkup and you've got some HPV-16 positives in your cervical swab, you don't need to be too worried about this, because at each stage you'll have most women will eliminate the virus. Okay, so it's only a minority of infections that will progress to a high-grade lesion and then progress from there to cancer. Now, which not all HPVs have an equivalent cancer risk. So if you look at the prevalence of the different alpha HPVs in cervical samples with an abnormal cytology, then you'll see that HPV-16 is the most prevalent one, 62%. Then HPV-18, then HPV-45, then 33, then a bunch of other ones. But if you look at the prevalence of these same viruses in samples that are positive for some HPV and for some human papillomavirus, but have normal cytology, then these high-risk viruses, they're really a minority amongst the human papillomaviruses that are in circulation in the population. Okay, so this prevalence here is kind of indicating the viruses that are around in the primary infection. Okay, samples that are positive for viral DNA, but with normal cytology, and this is what you've got in the cancers. So these are the dangerous ones, but they're a minority of the viruses that are circulating. Now, during this progression, once again, there are risk factors for progression from primary infection or a low-grade lesion to cancer. And the risk factors are number of full-term pregnancies. The more full-term pregnancies you have, the higher the risk of progression with a low-grade lesion to cancer, smoking, and HIV. So once again, it's immunosuppression is not good. So what are the therapies available? So if you've got cervical intraepithelial neoplasia stage 1, the low-grade lesion, where you'll be, observation, come in for a checkup, 6 months, we'll see what happens. Because most of these will resolve spontaneously. There's no need for therapy. Higher grade lesions, in 2 or 3, then you get some kind of surgical procedure, depending on where you are, depending on the recommendations for different countries. Colonization, LLETZ, hang on, I better read out what this is. So this is large loop excision of the transformation zone. Some surgical removal of the lesion or cryotherapy. If you've got an intra-cervical carcinoma, well, then you're in cancer care, and the therapy depends on the stage and what the oncologists say. It's not really an antiviral thing. So what's happening from a virological point of view during this progression? Okay, so the primary infection is down here. It's in the basal epithelial cells. Okay, so you've got these infectious particles from your sexual partner who's going to infect you with these HPV particles. And in these basal epithelial cells, there'll be just a restricted expression of small numbers of the viral early genes. And the viral genome will be restricted to 50 to 100 genome copies per cell. So there's maintenance of the virus in a kind of semi-latent state down in the basal epithelial cells. However, the E6 and E7 proteins will activate the cell cycle and provoke proliferation in the infected cells. And that's necessary for the virus, because this is a small DNA virus. It doesn't have any DNA of its own. it requires the cellular DNA polymerase and all the cellular cofactors for DNA replication in order to replicate this genome. So if it wants to amplify the viral DNA, it's got to push the cell into the cell cycle. And that's the role of these E6 and E7 proteins. So these kind of latently affected cells, as they differentiate, they'll express more of these early proteins, E6 and E7. They'll push the cell into the cell cycle. And then this allows amplification of the viral DNA, late gene expression, and then assembly and liberation of the particles. So the full application of the active viral replication is really restricted to these like upper layers of the epithelium. So these are further away from the blood and where you're getting some kind of immune surveillance in the mucosa. Whereas at the basal cells, which are more in contact with immune surveillance, there's not very much viral gene expression. So the whole replication cycle is kind of based around reducing the exposure to the immune system with only having actively replicating virus-infected cells right at the top here. So that's okay for the virus. The virus loves this. And you can have lesions that have a continuous infection for replication for months or years. And it's the same thing with low-risk beta papillomavirus infection on your skin. These beta papillomaviruses cause warts on the skin, and you can have them for months with replication restricted really to the upper layers of the epithelium. Now, the problem occurs once you have partial integration of the HPV genome into the host cell chromosomes, and specifically if you've got integration of the E6 and the E7 proteins. And then you've got this these viral oncogenes that are activating cell division, inactivating host cell apoptosis, and that's what gives you these high-risk lesions. And over the multiple years of continuous cell division, then these cells incorporate more and more somatic mutations in cellular oncogenes, and that's what leads to invasive cervical cancer. So that's a kind of model, but integration is not observed in all cases. Maybe the majority, I think about 2/3. But there are a significant minority of cancers where the viral genome is not integrated. But it's still there, and it's the function, the expression of the E6 and E7 oncoviral proteins is really essential for the carcinogenic process. Okay, how about the immune response? So now once again, viruses have got to dodge the innate immune response, and for papillomaviruses, it seems like TLR9 is particularly important. So in some experiments from about 10 years ago now, this was done by Massimo, done by a group in the IARC in Lyon. So they infected these primary epithelial cells with pseudotype virus or quasi-virus. Now, virologically, this is kind of interesting here because human papillomaviruses are not easy to culture. So it hasn't really been possible to get real replicative viruses. So what you can produce are pseudotype viruses. So the capsid proteins are present, but inside you'll have some kind of reporter gene. But the quasi-virus, this has got the capsid proteins and a kind of artificial viral genome that can replicate. Okay, it's got all the early proteins. So if you infect epithelial cells with a pseudotype, HPV 16 pseudotype, you don't get any change in TLR9 expression. But if you infect with a quasi-virus, then TLR9 expression is eliminated. And these cells which are infected cannot respond to CpG oligodeoxynucleotides. So TLR agonist, which is added here, in the infected cells, you don't get any interferon response. And that's confirmed in biopsies of cervical cancer. So now, what am I looking for here? So this is a kind of like normal epithelial tissue. You've got TLR9 expression, and in the cancer biopsy here, there's nothing. Okay, all of these cancer cells are HPV-16 positive, no TLR9. Now, other bits of information that kind of consistent with this is the observation that there's a correlation between TLR9 genetic polymorphisms and different cancer phenotypes. And also, kind of more slightly interestingly, is that bat papillomaviruses, not HPVs, because H is for human, right? Bat papillomaviruses co-evolve to avoid detection by TLR9. So this kind of TLR9 papillomavirus thing seems to be generally true amongst other animal species, not just in humans. Okay, what about humoral and cellular responses? So for humoral responses, as you all know, there's a very effective vaccine available against HPVs. The HPV genotypes act like serotypes. There's not that much cross-neutralization between different genotypes. But there is a bit, okay? So the data here is looking at neutralizing titers against the different HPV genotypes in a cohort of women vaccinated either by Cervarix vaccine, bivalent vaccine against HPV 16 and HPV 18, or the Gardasil 4 vaccine, which incorporated HPV 6, 11, 16, and 18. So red are the neutralizing antibody responses in women after Gardasil 4, and blue is the neutralizing titer in women vaccinated with Cervarix. So you can see, you know, pretty good neutralizing response against the vaccines, against the HPV genotypes present in the vaccine. But you've also got a bit of cross-neutralization against some of these other high-risk HPVs that are not present in the vaccine. So HPV 31, you get a bit of cross-neutralization. It's close to HPV 16. HPV 45, a little bit of cross-neutralization. And there's some evidence of cross-protection, okay, also against the cancers induced by these other high-risk viruses. However, okay, the thing you have to remember is that antibodies are protective against the primary infection. They don't have any action against progression of an established lesion, because the cancer progression is dependent on the early proteins, E6 and E7. It's not dependent at all on the capsid antigens, which is what the vaccine is made of. Okay, so how about the T-cell response? So first indications that you get some T-cell response come from, you know, the end of the second half of the 1990s. And then really pretty early on, everyone is interested in finding out whether the women with cancer have a CTL response or they don't. So this is maybe the first paper that goes along in this kind of sense. 1997, you get CTL responses detectable in women without lesions, but with positive HPV 16, but not very often in women with lesions. Similar kind of thing. This time T helper responses less frequent in women with carcinoma compared to women with only high-grade lesions. And then more convincing data with a kind of bigger cohort of women really starts to become convincing around about 2000. So here's 51 women with HPV 16 infection. They detected CTL in 22 out of 40 women who cleared infection. So this is a longitudinal study following women to see whether they clear the infection or whether it's maintained. But the women who had a persistent infection, none of them had CTL against the E6 protein. So the persistent infection correlates with the lack of the E6 CTL response. And this was confirmed in a vulvular intraepithelial neoplasia, another type of cancer associated with HPV-16. So there's this clear data from a number of studies that, lack of a CTL response is what is related to the persistence of the virus. Okay. So once you figure that out, then the idea is, why not have some kind of therapeutic vaccination? Try and induce this CTL response as a therapy for women who've got a low-grade lesions, or maybe even high-grade. So this started out, end of the 1990s, and then from about 2000 to 2010, 2015, there were a lot of different therapies that were tried, DNA vaccine, peptide, recombinant protein, RNA, viral vectors, everything. Now, these studies, they take a long time to go from a kind of concept to clinical trials. So this is one example here. So this is the first, proof of concept study we're showing immunogenicity in mouse of an RNA-based vaccine by Toos Darman in Holland, published in 2000. And the phase 1 clinical trial was only published in 2021. So 20 years of, preclinical development just to get to this stage. And probably the most encouraging data on this kind of trials published almost 10 years ago now, 2015. Phase 2B randomized controlled trial of a DNA vaccine, which, according to the abstract, was the first therapeutic vaccine to show efficacy against, high-grade lesions SIN2-3 associated with HPV-16 and 18. So I want to take a little bit of a closer look at this one, to see exactly what the level of success was in this trial. Okay, so here's the protocol. So they've got a randomized controlled trial, women with a cervical intraepithelial neoplasia, stage 2 or 3 lesion with HPV-16 or HPV-18. They got DNA vaccine, 3 doses versus placebo. So 167 versus 42. And the success is if the lesion regresses to SIN1, so it becomes less abnormal or normal. Okay, that's what you want. Or clearing HPV DNA. So in the modified intention to treat analysis, so the numbers here, these are basically women who had at least 1 injection, as I understand it in the protocol. So they had, in the vaccine arm, 48% of women had a regression of their lesion compared to only 30% of women in the placebo, in the control arm. For clearance of HPV DNA, almost 40% in the vaccine arm, only 15% in the placebo arm. So that's pretty good, right? It's working. You're giving this therapeutic vaccine and you get some kind of a real response higher than in the control arm. But that was 9 years ago now. So how come it hasn't been adopted as standard therapy? Well, one of the reasons is, this is, one of the reasons. The big reason is it's not effective enough. Okay, so this is a systematic review of all the different HPV therapeutic vaccine trials. This one here, Trimble from 2015, kind of representative of other types of vaccine modality. And overall, the average is you get about 54% regression of the lesions in the patients, the women in the vaccine arm. And that's only about 50% higher than spontaneous regression. So that's what this, risk ratio is here. So you've got 1.5, it's about 50% better than doing nothing. And that's not favorable compared to surgical intervention. So if you've got one of these surgical procedures, then your cure rate will be much higher than 80%. So that's why it hasn't really made the mainstream, okay? So, okay, it kind of works, but it's not efficient enough to replace the standard of care. It's less effective than standard of care. Now, there's some kind of hope for this because when they analyzed the women who were in the vaccine arm in this trial, the Trimble paper, then the women who had regression and clearance, they had a higher CTO response than the women who didn't clear the virus. So there's maybe some hope that if you've got an improved vaccine, then this might lead to a better clinical response rate. And so I think there'll be more things done on this. I know BioNTech, they've got vaccine trials going since they've got a huge stack of money from the COVID-19 vaccines. They're doing everything with RNA vaccines now. So I think this one is it doesn't work yet, but people still think this can work. And of course, if you manage to get a therapeutic vaccine that has equivalent efficacy to standard of care, then it might be worth doing, because these surgical interventions, mostly they don't have any impact on sexual or reproductive health, but there are some adverse events, of course. So if you can avoid them, then that would be good, but the efficacy has got to be good enough. Okay, so we've got about 15 minutes just to do herpes viruses, so I'll try and speed up a little bit. Okay, herpes viruses. So we've got big DNA viruses, envelope viruses, icosahedral capsid, and here we've got big DNA genomes, 120, 235 kilobases. Now, all of these viruses are able to initiate a latent infection. In a latent infection, viral DNA will be persisting in the infected cell as a circular episome, and most viral genes are not going to be expressed. So these latently infected cells are generally kind of invisible to the immune system. Now, herpes viruses, the orthoherpes viridae family, is divided into 3 genera, or in fact, no, subfamilies, in fact, the alpha, beta, and gamma herpes viridae. And the human herpes viruses, alpha herpes viridae, herpes simplex virus 1 and 2, varicella zoster virus, beta herpes viridae in humans, the most important one is cytomegalovirus, and the gamma herpes viridae, Epstein-Barr virus, and Kaposi sarcoma herpes virus. So the gamma herpes viruses are the ones that are associated with cancers. Okay, just a quick look at alpha herpes virus pathologies and therapies. So HSV causes oral herpes, about 70% seroprevalence in France. Most people get infected during childhood. I'm not really sure that everybody has a latent infection, but it can remain as a latent form in neurons that are innervating the site of the initial infection. And then you get recurrent lesions due to reactivation. Now, these primary infections are mostly quite benign, but in about 1 in 200,000 infections, you can get a very, very severe HSV encephalitis. So it's a rare occurrence. So herpes viruses, they're quite autonomous for their DNA replication. They have their own DNA polymerase. They have their own thymidine kinase. And these are the pharmacological sites of these antiherpetic antivirals. So acyclovir is active against the alpha herpes viruses. Varicella zoster virus causes chickenpox, varicella. About 90% seroprevalence in France. Most children, uncomplicated infection. And once again, you can get some rare neurological infections of VZV, of varicella virus. So it's got some kind of neurotropism. And then in older adults, it can cause a zona. So painful skin lesions can also be associated with neurological symptoms. Treatment, once again, these nucleotide analogues, acyclovir or famciclovir. And interestingly, there are effective vaccines for varicella zoster virus, an live attenuated vaccine and a subunit vaccine based on the glycoprotein E, envelope glycoprotein. So in France, these are licensed as prevention for zona, okay, herpes virus shingles. In other countries, there are attenuated vaccines licensed for use in children, and the subunit vaccine is more effective. So this is the one that is, prescribed now and recommended, I believe, adults over 60 or 65 years old. Okay, beta herpes viruses, CMV, cytomegalovirus is the most important one. Once again, primary infection in childhood gives a kind of flu-like syndrome or a mononucleosis sometime, and then can remain latent in myeloid progenitor cells. So it's in bone marrow CD34 positive cells found in monocytes in people with who are seropositive. Now, most of the time, doesn't really cause any problems in people who are CMV seropositive, but it can cause problems if you are immunosuppressed with, clinical AIDS, or if you're a transplant recipient and you're under immunosuppressive therapy to prevent rejection of the organ graft. So then you can get a kind of systemic viral infection with different types of symptoms. And this is treated once again with, these nucleoside analogues, not the same ones as for alpha herpes viruses. Now, the other big problem is maternal-fetal transmission of the virus. When a pregnant woman has a primary infection and there's high viral load in the with active viremia. So the virus can cross the placental barrier and infect the fetus. And this, well, most of the time will be the baby born will be okay. But in a minority of cases, unfortunately, maternal fetal infection will cause deafness, mental problems, and in severe cases, very, very severe pathology or death just after birth. So maternal fetal or intrauterine CMV infection, I think it's the #1 cause of congenital deafness in France. So this is one of the big issues. And it's the primary infection in a pregnant mother which is the big risk factor here. So women who are at risk is their second or third pregnancy. Okay, they've already got a kid in the crèche or in the primary school, and the first child is getting infected with the CMV and bring it home, and then that will transmit the virus to the mother. So that's the kind of profile of a high risk for maternal fetal CMV infection. Okay, Epstein-Barr virus. What do we got here? Okay, so once again, almost everybody's getting infected during childhood. The younger that you are, the less severe the symptoms are. So if you get childhood infection, most of the time, they'll be asymptomatic or with a few symptoms. If you get infected as a young adult or an adolescent, then maybe 30 to 70% of these infections will give you an infectious mononucleosis. So the virus is going to be replicating in the oral mucosa, in the mouth, and transmitted by kissing. That's why you've got this peak of transmission in young adults as they become sexually active. Now, it establishes latent infection in memory B cells. And it's got a lot of interesting disease associations with autoimmune diseases. So there's good epidemiological data that reactivation of these latent EBV infections in B cells seems to be related in some way to the onset or the exacerbations of systemic autoimmune diseases like lupus, rheumatoid arthritis, Sjogren's syndrome, this kind of thing. And MS, multiple sclerosis, sclerose en plaques in French. The other big pathology related to Epstein-Barr virus are cancers. So Burkitt's lymphoma, Hodgkin's lymphoma, diffuse large B-cell lymphoma, post-transplant transplantation lymphoproliferative disorder, and some epithelial cancers. Not so much in Europe, but it's a cause of nasopharyngeal carcinoma, and also 10% of gastric cancers. So there's no real antivirals against EBV. Okay, so what are the innate immune effectors that detect herpes virus infections? So as you might imagine, they are the PRRs that are detecting viral RNA or DNA, viral nucleic acids. So for DNA, TLR9, cGAS-STING, we know about that. IFI-16 is an interesting one. Okay, so this is mostly kind of present in the nucleus. It's an intranuclear pattern recognition receptor, binds double-stranded DNA, but it can also shuttle from the nucleus to the cytoplasm. So alpha and beta herpes virus DNA detected in the cytoplasm of myeloid cells by IFI-16, and gamma herpes virus DNA detected by IFI-16. RNA, also interesting, very important. So TLR3 is an important PRR for herpes simplex 1. And we know this because rare mutations in TLR3 are associated with herpes simplex encephalitis. So the inability to detect the virus with TLR3 leads to a much more severe infection. Now, another interesting one is RIG-I. And this is also kind of weird because these are DNA viruses, right? So you think, why is RIG-I, which is going to be activated by unusual RNA structures in the cytoplasm, why is that detecting a DNA virus infection? Well, it seems to be indirect. So during herpes virus infection, then this host cell RNA polymerase 3 transcript, which is a kind of 5S rRNA pseudogene, will leave the nucleus, be translocated to the cytoplasm, and activate RIG-I. So RIG-I activation isn't really detecting anything that's specific for the virus. It's somehow detecting aberrant nuclear transport of nuclear cytoplasmic transport of RNAs. So this is a kind of like alarm signal that the cell is giving to say something's going wrong in the nucleus. And the last one, which is also very surprising, is the role of TLR2. And there are a lot of papers that show that herpes virus glycoproteins can activate an inflammatory response via TLR2, which I always find very surprising and almost hard to believe. But there's a lot of data on this. TLR2 is supposed to be like bacterial glycolipids, isn't it? Well, why are viral proteins activating this? And I think the precise molecular interaction really isn't known. Okay, so how do herpes viruses dodge these innate pattern recognition receptors? So one of the things, they've got big genomes, they've got lots of genes doing a whole bunch of things to inactivate or dodge the immune system. So I'm not going to talk about everything. I'm just going to present some things that are specific to herpes viruses. And in particular, herpes viruses have these things called tegument proteins. So the tegument proteins are found in between the capsid and the viral envelope. That's all these things kind of in light blue cyan here on the diagram. Now, what is interesting about the tegument proteins is that when the virus enters the infected cell by fusion at the plasma membrane, so you imagine this virus envelope will fuse with the cellular plasma membrane, all these tegument proteins are released into the cytoplasm of the infected cell right at the very start of the infectious process. So they can go in and kind of immediately disrupt host cell responses. So here's a few of these effectors that I just talked about in the last slide. cGAS-STING, RIG-I, and IFI-16. So some tegument proteins will target IFI-16 for degradation or inhibit it. And other tegument proteins can bind to and inhibit cGAS. Another one will be inhibiting STING. HSV US3 prevents IRF3 activation. And there's a very interesting tegument protein called UL37, which can bind to and deamidate critical amino acid residues in cGAS and RIG-I. So this one tegument protein is activating both these branches of the, cellular pattern recognition receptors that are going to be able to detect these viral nucleic acids. So there's a very, very important role of the tegument proteins of these viruses in inactivating the innate response. Okay, T cell responses. I'm going to be running out of time here. Can I run over 5 minutes? Next one is 10:45, right? And I started 3 or 4 minutes late, so I guess that counts. Okay, so the general kind of picture that emerges looking at these different herpes viruses is that the immunodominant viral antigens for T cell antigens or immunodominant T cell antigens are really either the tegument proteins, so they're delivered into the infected cell immediately and they can be processed and presented on HLA class 1 very, very quickly, or they are the early genes, the immediate early genes, IE1, IE2, which are the first genes expressed in the infected cells. So these are very, very good CTL antigens because they will allow the CTL to lyse the infected cell very, very early in the replicative cycle of the virus. So these are the immunodominant T cell antigens in the lytic infection. However, in the latent infection, these major T cell antigens aren't really expressed. And so what seems to occur is that periodically there's reactivation of a lytic infection cycle, and then these newly reactivated infected cells are once again recognized and controlled by memory T cells. And as this is repeated over your life, then there's a subset of the T cells that are engaged during the primary infection that are getting re-stimulated time and time again over your life. And this seems to select for the high affinity T cell clones, which are expanded and come to dominate the T cell repertoire in people who are seropositive for these viruses. And that's a phenomenon called memory inflation. You've got a small number of clones that are expanded and they dominate the T cell response. So that seems to be good enough to control the virus until you have a waning of the T cell response. Either you get older, immune senescence, or you have some kind of cause of immune suppression, and that's what leads to a symptomatic reactivation. So what T cell responses are important for controlling reactivation? So one of the types of immunosuppressed states is if you have an organ transplant, and once again you'll be on immunosuppressive therapy, and this can allow herpes viruses to reactivate. So this was a study comparing CTL against CMV, the two immunodominant antigens, IE1 and PP65. And so the patients who did not have any CMV disease had an IE1 response. And this has also been replicated in other studies since 2005. That's almost 20 years ago now. So this is a well-established observation. And what this means is that T cells against IE1, but not against PP65, have a role in protecting against symptomatic CMV reactivation. And that kind of makes sense because IE1 is the first viral protein that's going to be expressed in a latent cell which is reactivated. Whereas PP65, it's a late gene, it's a late protein. So it's not so good to have a CTL directed against this late protein in a reactivation scenario. One of the kind of consequences of this memory T cell expansion is that people who are CMV seropositive have a lot of their memory T cells, which are basically directed against cytomegalovirus. And this ends up having an impact on the overall composition of PBMC subsets in normal, healthy people. So this was a result that was, I think quite surprising, came out of the Milieu Intérieur Consortium at the Pasteur Institute, which is led by Louise Quintana-Merci, Dara Duffy, and Matthew Albert. So they followed like 1,000 people, had a standardized phenotyping protocol for PBMC subsets, looked at a lot of genetic factors and environmental factors. So the 4 big environmental factors have an impact on your circulating PBMC subsets are age, smoking, sex, and CMV serostatus. So CMV, latent CMV infection, it's as big an impact as being a smoker. So that's quite a significant thing. And specifically what they found is that people, these aren't patients, these are healthy people, normal people. They have a bigger number of CD4 and CD8 TEMRA subsets. So TEMRA is this effector memory RA positive cells, which are CCR7. What's that? CCR7 positive? No, CCR7 negative, right? Oh, I forget. Okay, it's some T cell thing, right? But memory cells that they always go on about them being memory cells. I never worked out why they aren't just effector T cells, right? Well, anyway, probably people in the lab will be able to explain it to me once. Okay, so people who have got are CMV seropositive, they've got quite a large proportion of their memory cells taken up by these anti-CMV T cells. Now, does this actually have any functional impact on the response to new antigens? Because as you get older, of course, you've got fewer naive T cells. And maybe the best result here is like published just a couple of years ago with Francesco Nicoli, together with Victor Appé in Bordeaux, when Francesco is down in Italy. So they studied the response to a vaccine in a cohort of patients more than 70 years old. The interesting thing about this is the vaccine is against tick-borne encephalitis virus. So nobody in this cohort has seen this antigen before, okay? It's not like influenza, it's not like CMV or something like that, not like tetanus. So this is a primary immune response. And so they found that, in fact, the older people who were vaccinated, the people who were CMV seropositive, had lower T cell response and a lower humoral response. So this persistent chronic CMV infection is actually having a bit of an effect on the T cell response. Last thing about T cells and CMV and herpes viruses is that I didn't want to go into this, but herpes viruses have got a whole bunch of mechanisms to inactivate or avoid presentation of CTL epitopes in infected cells. So I think this will kind of result in non-conventional T cell subsets and NK cells being a bit more important in the control of these viruses. So in CMV, there's an expansion of gamma delta T cells in kidney transplant recipients with CMV. So this was observed 25 years ago, Julie Deschanel-Merville in Bordeaux. And the expansion of the gamma delta T cells was correlated with reduction in viral load. So they seem to be controlling the CMV. Since then, a bunch of the mechanism has been elucidated a little bit. So the CMV infected cells seem to be expressing these kind of gamma delta T cell ligands, which are some kind of indicators of cell stress. And there's something similar in Epstein-Barr virus, but this time, in fact, it's a different gamma delta T cell subset. And the V gamma 9, V delta 2 gamma delta T cells, these are the ones that recognize pyrophosphate phosphoantigens presented on these butyrophilins BTN3A1. And these gamma delta T cells seem to be able to recognize latently infected EBV cell lines. So there's a kind of role for gamma delta T cells in the responses against these viruses. Okay, last couple of slides here. Actually, it's the last one. So we're on to the herpes viruses and humoral responses. So there's no real evidence for superinfection of herpes viruses. If you've got HSV1, you've got a latent infection, but there's no real evidence for a second superinfection. So that probably indicates that antibodies are protective against, the primary infection. And of course, the effects of, however, there's no protective effect of antibody, or not very much, against reactivation. So in Epstein-Barr virus, the infected B cells, are proliferating. They're not really liberating new particles. It's just the latently infected cells that are proliferating. In the VZV, and the efficacy of the vaccine seems to be correlated with the CD4 T cell response. And once again, HSV1, HSV2, and CMV, cell-to-cell transfer of virus seems to be very important. Not so much the effect of free virus infecting new cells. So just to sum up on the last slide here, so hepatitis C, you've got subversion of the interferon response, CTL escape, glycan shield, and cell-to-cell spread. HBV, it's kind of a stealth infection, not much activation of the interferon response. HBs antigen seems to induce specific immune tolerance. HPV's TLR9 is the most important pattern recognition receptor. Humoral response, antibodies prevent the primary infection. They don't prevent or have any effect on the persistence or the progression to cancer. And then E6 and E7 specific T cells are the ones that are responsible for clearing infection. And herpes virus is a whole load of different things. One of the things that we need is a vaccine for CMV. Why? Because it could be useful to prevent this maternal fetal transmission, or it could be a preventive vaccine in patients who are going to have an organ transplant. So I'll just finish up there, and I'm sorry about going over my time. Thanks.